Hi all of you. Today in this session we are going to see what are antibiotics and introduction to their antibiotics along with their classification. Coming to antibiotics, antibiotics are chemical substances which are released by living organisms and which are used to kill some other microbes. For example, penicillin. It is released from fungus Penicillium notatum and it is used to kill bacteria. So the word antibiotic can be divided into two parts. Anti means against, biotic means life. These are the substances released by living organisms against the life of other species. So myself, Mr. Nagaprashant. Please subscribe to our channel for regular updates. The word antibiotics was introduced and coined by Paul Ehrlich. He called them as magic bullets because these antibiotics are very specific to pathogens, not to normal living human cells. So these antibiotics will not produce any effect on human cells, but they will kill only pathogens, maybe fungus, bacteria, protozoa, virus, etc. That's why those are called as magic bullets. If they were given a target, they will reach at a specified focus. That's why those are called as magic bullets. And the person behind the term antibiotics is Paul Ehrlich. A legendary person for today's antibiotics, Alexander Fleming. He identified and discovered the presence of penicillin in a fungal strain called Penicillium notatum. While doing his microbiological experiments during Second World War, he identified the presence of penicillin in a mold. And later, the production of penicillin started at industrial scale and it became a revolution in entire world for treating infectious diseases. So what is the need for the study of antimicrobial agents or antibiotics? There are few terms or few categories or few criteria to be known before studying these antibiotics. There are 6 to be very specific. First one is selectivity. You see, antibiotics have selective toxicity towards bacterium rather than the host. That's what I told in my first slide itself. That's why those are called as magic bullets. Whereas disinfectants lack selectivity, they will kill all cells, all viable cells. Higher the toxicity, lower the selectivity, or vice versa. Higher the selectivity, lower the toxicity. Therapeutic index. The drugs with better therapeutic index are called as, or the drugs with higher therapeutic index are called as better antibiotics. And categories of antibiotics can be either bactericidal or bacteriostatic. So depending upon the seriousness or intensity of the infection, we have to choose either a bactericidal antibiotic or bacteriostatic antibiotic. Bactericidal means our given antibiotic is going to kill the bacteria. Cidal means killing. Bactericidal means the compound which kills bacteria is called as bactericidal. And coming to bacteriostatic. Static means stopping the growth. If the process of multiplication of number of this bacteria was stopped by stopping its reproduction or growth, then that is called as bacteriostatic. In the same case, fungicidal, fungistatic. in case if it is showing effect on fungi. So like that. And serious infection should be treated with bactericidal antibiotics, whereas if the infection is in starting state, we may opt for bactericidal. And antibiotic susceptibility testing. The effect or efficacy or the potency of the antibiotic can be assessed by doing these two tests: minimum inhibitory concentration, minimum bactericidal concentration. The minimum inhibitory concentration is the minimum concentration at which it can show its effect to kill a bacteria, whereas minimum bactericidal concentration is the minimum concentration at which it is killing the bacteria. Okay, so the MBC, that is minimum bactericidal concentration, is lowest of antibiotics that kill 99% of inoculum in the given time. So combination therapy, so as we know, synergism and antagonism. So in general, antibiotics are used in a combination like mixing bacteriostatic and bactericidal agents. In combination, they will show very great effect. And last one, antibiotics and chemotherapeutic agents. Antibiotics strictly refers to the substance of biological origin. So as I told, antibiotics are originated or produced from a living organism and which are used for treating or killing other pathogens, whereas chemotherapeutic agents are synthetic chemicals. So you see here, apart from these two, semi-synthetic drugs are also there, semi-synthetic antibiotics are also there, which means we are doing slight structural modifications to the naturally available antibiotics. Then those are called as semi-synthetic antibiotics. Coming to natural antibiotics, penicillin V, penicillin G are best examples. And coming to semi-synthetic, amoxicillin, ampicillin are best examples. And chemotherapeutic agents, that is purely synthesized compounds, fluoroquinolones and sulfonamides are best examples. So with this, these are all the points to be considered while choosing an antibiotic for the treatment. Discovery timeline for these antibiotics. You can see the timeline has been divided into two: post-antibiotic era and pre-antibiotic era. Okay, post-antibiotic era from 1990 to till date, not even a single new antibiotic has been discovered. All antibiotics what we are using today are discovered before 1990. From beta-lactam around 1928, beta-lactam, that is penicillins, were discovered by Alexander Fleming, followed by sulfonamides, aminoglycosides, cycloserines, tetracyclines, macrolide antibiotics, monolactam, fluoroquinolones, and at last lipopolypeptides. All these antibiotics were discovered before 1990, and later still research is going on to identify the best suited antibiotic. So best suited means which is having good potency, which will not develop resistance, and which shows good effect at lower concentration, which is having less ADR. So identifying an antibiotic with such a great quality is really a great task for scientists. So the search is still going on for the best antibiotics. And coming to the classification based upon their structure, or it can also be called as a chemical classification. Beta-lactams, in which you can find 4 subdivisions like penicillins, cephalosporins, monobactams, and carbapenems. Later sulfonamides and trimethoprim. In combination it's called as cotrimazole. Aminoglycosides, tetracyclines, chloramphenicol, macrolides, glycopeptides, quinolones and fluoroquinolones, phosphonic acids. You can see their general structure like beta-lactams, a four-membered ring with nitrogen azepine with thiazole, sulfur and nitrogen, a five-membered ring. A combination of this ring is called as a beta-lactam. So this beta-lactam ring is a basic requirement for its activity, followed by sulfonamides. The structure of the sulfonamide is very similar to that of PABA, para-aminobenzoic acid. That's why it is showing its activity as antimetabolite, followed by aminoglycosides. These are carbohydrates which are linked up with glycosidic linkages, and you can find glucose like moieties here. Tetracyclines, which are having four cycles side by side, chloramphenicol, and a macrolide. Macrolide means, macro means big, lide means lactone rings. So macrolides are large lactone rings. Glycopeptides and streptogramins, quinolones, oxazolidindiones. These are the various chemical classes of antibiotics based upon their structure. And next, coming to the classification based upon their mechanism of action, basically they can be divided into five. The antibiotics which are affecting the protein synthesis, which are affecting nucleic acid synthesis, which are affecting the metabolic process of antimicrobials, that's why those are called as antimetabolite antimicrobials, which are acting on cell wall and which are acting on cell membrane. Here you can see the mode of antimicrobial action, and here it's very clear. Cell wall synthesis inhibitors like cycloserine, vancomycin, carbapenems, cephalosporin, penicillin, monobactams, etc. Folic acid metabolism inhibitors, that is antimetabolites like sulfonamides and trimethoprime, and cell membrane inhibitors, polymyxin. You can see DNA gyrase and quinolones, which are going to affect the synthesis of nucleic acid or DNA. Protein synthesis and all are affected by these ribosome and 50S and 30S ribosome, like protein synthesis inhibitors, 50S inhibitors or erythromycin, chloramphenicol, whereas 30S inhibitors or tetracycline, streptomycin, gentamicin, kanamycin, etc. So these are all various classes of antibiotics based upon their mechanism of action. So apart from this very important topic which we should need to discuss is resistance. Nowadays this resistance is really annoying the humankind for treating many infectious diseases. So resistance will be of three types, like clinical resistance, cross resistance, and multiple resistance. And these all types of resistance will inhibit the treatment of the infectious diseases or killing of these pathogens, because these pathogens will develop a sort of resistance towards the given antibiotic. So the treating of tuberculosis, for example, is a very best example, which became very difficult for treating. And mechanism of resistance, five possible mechanisms are there. Altered permeability of antimicrobial agent, so that the antimicrobial agent or antibiotic cannot enter inside the cell of pathogen. Inactivating the antimicrobial agent, immediately they will develop an enzyme which will break down the structure of the antibiotic so that it cannot show its activity. Altered target site. The target site, that is either an enzyme or a ribosome or an DNA gyrase, whatever it may be, changing its structure by itself by doing some modifications towards its genome, then our target cannot be achieved because of change in the shape or whatever it may be. Replacement of sensitive path, like for example, preparation of folic acid from PABA. If you are blocking one enzyme, then the pathogen may try to synthesize the same folic acid through another route, and excretion or exclusion of these antibiotics. So whenever our antibiotic tries to enter into the pathogen, immediately it will develop an mechanism or pumps which will send them outside the cell with a force. So these are the possible ways of mechanism resistance. So with this, I would like to close today's session. So in our today's session, we have gone through the definition of antibiotic, and the person Paul Ehrlich who identified and coined the term antibiotic, the person, another scientist, Alexander Fleming, who was the first to discover an antibiotic, that is penicillin. After that, we have gone through the requirements and purpose of studying these antibiotics, and later we have studied two types of classifications. One is based upon structure, another one is based upon mechanism of action, and at last we had a small insisting about resistance of these pathogens towards the antibiotics. So if you have any doubts in this slides and video, I would feel happy if you can comment in the chat box. Thank you.